Hello friend. In this module, we are going to see the application of statistics during a pharmaceutical product or pharmaceutical research and development. Statistics or biostatistics is being used at various stages of the pharmaceutical research and development. We will see what are the stages and what extent the statistics is used during the pharmaceutical research and development. Throughout most of the 20th century, pharmaceutical regulations was largely a matter of reactive legislation. Reactive legislation means if there is any issue or a problem with respect to the product or the packaging system or some other issues, quality issues, the legislation changes or the laws changes to overcome the issues. But in the 21st century, we have seen the rise of science-based regulatory initiatives, including ICH guidelines on pharmaceutical development, pharmaceutical quality systems, FDA guidance on GMP, good manufacturing practices, process analytical technology, and quality by design. These initiatives have brought a new emphasis on the process understanding and quality and the use of statistical methods for both. So basically what we are trying to say is that the statistical methods or the biostatistical methods are mainly used to improve the process and the quality of the pharmaceutical development. In pharmaceutical development, in discovery research, where the new drugs or new active pharmaceutical ingredient has been developed, the discovery process takes place in 5 parallel paths. That is the clinical trials, the API development, the preclinical assessment, then the drug product development, that is the final dosage form, then the analytical method development. These all the 5 process has been done parallelly, and each development process, each path requires specific statistical techniques during the process. We have seen that the discovery research goes on 5 parallel paths like development of API, development of finished product, clinical trials, preclinical assessment, analytical development. All this process uses different tools and statistical methods, which includes design of experiments, especially during the product development process, then the screening experiments, optimization studies, regression modelings, process optimization, and robustness studies. What is design of experiments? Design of experiments is generally used during the pharmaceutical product development, and design of experiments is a systematic method to determine the relationship between factors affecting a process and the output of that process. In other words, it is used to find cause and effect relationships. This information is needed to manage process inputs in order to optimize the output. For example, speed and length of a spironizer process within the studied range had a negative effect on the yield of desired quality of pellets, while the percentage binder and percentage granulation water showed positive effect on the yield of proper pellets quality. The negative effects of speed and length of the spironizer can be understood by the fact that high speed or the high length cause the pellets to stick together, making triplicates and quadruplicates. That means it forms the agglomerates, which itself takes the pellets to grow larger than the desired sizes. In continuation to the example, the granulation time of the pellets was selected based on the prior experience with the extrusion spiralization process. It seems that the selected granulation time range had a very small effect on the yield by not affecting the texture and consistency of granulated powder. So this particular parameter need not to be a critical parameter, but the spiralization time and the length may be a critical parameter in the design of experiments. During a drug discovery process, one of the parallel path development process is the analytical development. The analytical method development employs various statistical techniques, including the analysis of variance component studies, method ruggedness studies, other basic statistical techniques like the graphics and summary statistics. In the pharmaceutical manufacturing process, whether it is a discovery process or a generic process, the statistical process control, SPC, is used extensively to monitor and improve the process. For process improvement, statistical techniques found in Six Sigma, quality process, lean manufacturing, process analytical technologies, design for Six Sigma, and quality by design. What is process analytical technology? Act is defined by the FDA as a mechanism to design, analyze, and control pharmaceutical manufacturing process through the measurement of critical process parameters that affect critical quality attributes of an active pharmaceutical ingredient or finished good. The use of FACT enables users to establish a quality by design approach which aims to make quality product that is verified in real time rather than testing for quality after the process is completed. Quality by design. Quality by design is an approach that aims to ensure the quality of medicines by employing statistical, analytical, and risk management methodology in the design, development, and manufacturing of pharmaceutical products. Design of experiments and process analytical technology is part of quality by design. Design a formulation and identify the critical material attributes or critical quality attributes of the final product that must be controlled to meet the target product quality profile, and design a manufacturing process to produce a final product have these critical material attributes. So these are the elements which is important during a quality by design approach. During the manufacturing process, identify the critical process parameters and the input material attributes that must be controlled to achieve these critical material attributes of the final product. Use risk assessment to prioritize process parameters and material attributes for experimental verification. Combine prior knowledge with experiments to establish a design space or other representation of process understanding. Then establish a control strategy for the entire process. So these are also the important elements of the quality by design approach. So far we have seen the importance of statistics or adaptation of statistics, use of statistics in the pharmaceutical development process, which are parallelly happening like the pharmaceutical product development, pharmaceutical analytical development, and also the pharmaceutical manufacturing process. Then the other process is critical process is the clinical trial applications. So during the clinical trial studies, what are the role of statistics? The statistics is used for the designing of the studies, then the sample size estimation, the endpoint analysis, and the randomization. So when it comes to the clinical trial for the discovery of research, then it comes to the generics and the bioequivalence is part of the pharmaceutical development, where also the statistics is being used to design the bioequivalence studies during the generic product development, then the sample size estimation for the bio studies, then the endpoint analysis of the bio studies, then the randomization process during the bio studies. So friend, with this, we are concluding the module which talks about the application of statistics in various stages of pharmaceutical development. In the upcoming modules, we will see the application of statistics in clinical trials as well as bioequivalent studies and the basic concepts of statistical applications used for the clinical trial and bioequivalent studies. Thank you.